Hi everyone. Today I'm pleased to present a short preview of our new app, JBrowse 2. JBrowse 2 is a ground-up rewrite of JBrowse written in React that is made to address some new use cases. In particular, we wanted to make JBrowse more useful for people visualizing things like complex structural variants, and it allows you to have multiple views on the same screen at once. In this demo, we're going to look at a workflow that we call the Structural Variant Inspector. I think it will be helpful to just go ahead and get started. So JBrowse 2 is a desktop application, just similar to any other app that you would run on your desktop. On Windows or Mac, you'll be able to just double-click an exe to get it started. I have it open now, and it's showing the welcome screen. And on here, we have the ability to start these new sessions. The linear genome view is sort of your standard genome browser, and the structural variant inspector is this new sort of wizard to get an overview of your data that we're going to look at today. So let's click on that. So here you're given the option to open up a new file, and this is the file that contains all your structural variant calls. So for our demo, we're going to look at this data that was, say, given to us by a collaborator. This is actually from the SHOTS lab, the SKBR3 breast cancer cell line. This contains PacBio sequencing, Illumina paired end sequencing, and various structural variant callers on both of those. We're going to just go ahead and take this VCF file that contains all the translocation events and plug that in as the URL. So we paste that in. It automatically sees that it's a VCF, and we open that up. So what this view is a combination of a spreadsheet and a Circos view, where the Circos data is populated directly from the spreadsheet. And this can be really useful for quickly drilling into your data. Say that we want to just filter out the things that we're interested in. Maybe we're interested in events that are happening on chromosome 20. So we can quickly type, say, CHR2 equals 20, and that's going to do a quick text filter on all the rows and actually match the info field in the VCF. Now you can imagine a lot more filters, and it's definitely something we'll add as needed. But this narrowed down our Circos view to only contain these rows from our spreadsheet. And that really clarifies some things. And we can also turn on this option that actually only shows the relevant displayed regions or the relevant chromosomes in this set. So let's say just that we're also interested in events related to chromosome 20 and chromosome 9. So we can actually click on these little chords, and it'll pop up what we're calling the breakpoint split view. And this is basically two linear genome views stacked together, and it shows the connections between them when you show different data tracks. So let's open up, let's say, the Illumina BAM data on both of these and see what happens. So this shows that there's these pairs that are going from chromosome 9 to chromosome 20, and they're illustrated by these connections that are drawn across the screen, across the two views. And this is all interactive, and you can scroll around, and it should be fast and responsive to your inputs. Sometimes you end up seeing the sort of crisscross, and we actually allow you to horizontally flip the view to straighten things out. And that'll just generate more unoverlapped connections. But from this Illumina reads, we see a lot of evidence for this connection being made, but we're not seeing the sort of base pair precision that we can actually get with our PacBio sequencing. So let's turn on the PacBio data. I'm going to just close the Illumina data to make it clear. So this PacBio data, once it's loaded, has to chug through all these long reads. Really gives us a more precise picture of what's going on. We have this setting for drawing the connections that are, say, between a read on the same chromosome here. And we can, since we're only interested in the cross chromosome connections, turn that off and then see what's happening. Maybe we'll also want to turn on the VCF file that will show us what call it made. So after both of these views are turned on, I think we can get a lot clearer picture of what happens. So this VCF file is suggesting basically that this chunk in between here and here, indicated by the sort of feet on both ends, is basically inserted onto chromosome 20 down here. It just sort of grabs this chunk and inserts it. And all the evidence being available to you on this breakpoint split view really helps you sort of get a big picture of that event. Now, we can also turn on this setting, which actually allows us to interact with these little connections. So this guy are all clickable once you turn this setting on, and it'll give you all the details about this line from the VCF essentially. You can also do the same thing with the alignment squiggles, and it'll give you both ends of the reads details. So this is pretty much the main functionality of our breakpoint split view. You're able to additionally turn on any types of evidence, say, repeats that might help you understand more, gene track, additional ClinVar annotations that help you understand maybe the significance of this type of event. With that, I think we can maybe move on to a small other segue. As long as we're talking about all these evidence tracks, we have the ability to connect to other data resources that are on the web. Preset in our chipped version, we have a connection to the Cosmic Cancer data. Once you turn this on, it gives you all the features that are available from Cosmic, basically natively as tracks down here. So we can instantly get all the data that's from Cosmic right in our desktop genome browser. You can also do things like connect to track hubs that are out there on the web from the Track Hub registry or your own supplied links. With the Track Hub registry, we have this sort of interesting world of data at our fingertips idea, where once we look up all the track hubs that are available to us, we can instantly turn them on and connect to them. So let's say I'm sort of interested in the conservation of this. Typically that would be done for sort of a gene level annotation, but just for fun, we can turn on the phylo-CSF track or the phylo-CSF connection and get all the tracks that are available there. And really, it'll depend on your use case, your data, but that's the type of thing that we're trying to enable, is basically fetching any data that's of interest to you and contextualizing it in the genome browser. So there you have it. We have basically demoed our SV inspector workflow along with the breakpoint split view that is launched from that, and a couple other goodies like our track hub connections. I hope you enjoyed this, and we're going to send out some emails giving you all the links and data necessary for you to follow these steps yourself. And we welcome any and all feedback. This is all very new, and we're looking for more fresh ideas that help us help you better. So thanks a lot for watching, and I will talk to you later. Thank you. Bye.